it was quite a surprise to be asked, to be honest. But I'm delighted to be here. So today I'm going to talk a little bit. I'm going to tell you a little bit about the background of amphibian declines and why we care about counting frogs. And I'm going to share some survey methods with you that I have conducted over the years, and a new method that I developed about 5, 6, 7 years ago, and talk about how we, how we do that, and some possibilities of what we can do with that information. So you probably are very aware that amphibians are declining globally. At least 30% have been documented as that have been assessed by the International Union for the Conservation of Nature, have been identified as being threatened with extinction. And a bunch of amphibian species have actually gone extinct forever and ever, right under our noses. And of course, the exact numbers are hard to estimate because these animals live in remote areas. They're really hard to find. It's really hard to determine if something's actually there or not, or if you're just not seeing it or hearing it. You may know about the example of the Costa Rican golden toad that went extinct like in 1989. So fairly recently in our knowledge and understanding, and this was a highly abundant frog that people were kind of studying. And then one day it was just gone. And Dr. Joe Mendelson, who's a friend of mine, who works at the zoo in, I think he's in Atlanta, and he's started this program a number of years ago to save at least 2 individuals of each of these species, so like a Noah's Ark of amphibians. So frog populations are declining, and the reasons are various, right? Habitat destruction from loss of ponds and roads bisecting habitat corridors, pollution and chemical contaminants like atrazine, invasive species in the Virgin Islands mongoose, but elsewhere rats, cats, and even other frogs. Climate change that's contributing to increased drought and habitat conversion, UV radiation, over harvest. A really good example of that is the mountain chicken, Eleutherodactylus phalanx from Dominica in the Caribbean, pet trade, and of course the biggie disease, specifically chytrid. So we need to know more about our frogs. There's a lot of species out there. There's 6,000 and something. Every time I look, there's at least 20 more added. So there's a lot of frogs out there. The more we kind of are applying new methodology like genetic assessment, we're learning that what we thought was a frog is actually 3 or 4 different species of frogs. So there's a huge gap in our understanding of who these animals are, and we need to better understand things like community assemblages and habitats and the distributional range of these animals, which will tell us how sensitive they might be to environmental changes, and then that can give us direction for management so we can reverse some of these patterns. So I like frogs, right? I bet you'd like frogs too. They're charismatic, they're super cute, right? They're iconic. Think about Kermit the Frog that everyone grew up with. They're really critical ecosystem service providers. They eat a lot of bugs, they recycle those nutrients back into the environment. And they're really good indicators of environmental quality because they're sensitive to environmental conditions. So I'm actually a squamatologist, right? I studied lizards and snakes. And, Russ mentioned that I'm also a chiroptologist. I study bats. And everybody, you probably all know that feeling when you find a squamate, a lizard or a snake that's, they're just so hard to find, and it's so exciting when you actually see one. And so I'm going to give you a little chance to do some herping here. There's a lizard in this picture. Can anybody see it? Anybody find it? Russ says he can find it. Yeah, so this animal is there, right? There's its eye, there's this mouth, chin, and its body extends back here. This is a gecko in New Zealand, one of those really rare endemic geckos, and this is actually in a living museum collection. So it's super exciting to see something that's so cryptic. But I have a 25-year history of playing around with froggies. In the 1990s, I was a PhD student, and I was doing frog surveys for the Kent Reptile and Amphibian group that I co-started in England. And in the 2000s, I was working with the Declining Amphibian Task Force and with the Army, the Amphibian Research and Monitoring Initiative, and the National Park Service to document frogs across the Colorado Plateau. And then in 2004, I moved to the Virgin Islands, where I established a amphibian and reptile conservation program that entailed a lot of monitoring of frogs and distributional assessment and threats and stuff like that. And then in 2015, I started the cell phone frog project that I'm going to talk about today. And that's me as a young lassie in Brazil, marveling at the fact that somebody actually saw this teeny weeny little froggy. And we were able to actually look at it. Finding frogs, right, as opposed to finding squamates, is a lot easier, right? They're big, they stand out, they make a lot of noise, right? And so they're a lot easier to find. But depending on the habitat, it may be very challenging to actually get in and look for these things. And I can tell you from experience in the Virgin Islands, it takes me a good maybe half an hour to find a single individual frog, just to lay my eyeballs on them, not literally, just because the habitat is so cluttered. So you need another way of finding them. And something else that we, like about our frogs is, let me see if, oh, no, let me go back. Let me see if I can, my Zoom thing is in the way of the button and I can't move it. Well, the froggies make a lot of noise. Let me see, there we go, got that moved, right? So some of you may or may not have participated in the North American Amphibian Monitoring Protocol, where volunteers would be given cassette tapes, right, that they would listen to and memorize, and they would learn all the calls of all the frogs, and they would get in their cars at night and they would drive around a route that was given to them, and they would stop and listen for 5 minutes and write down what they heard, and they would collect some other environmental data like moon phase and precipitation and stuff like that. This program went on for a good, I don't know, maybe 10 years or so before it was halted. And I'm not really sure the reasons why it was halted, but it was a pretty cool program with a lot of people that got involved, and maybe their amphibian ID wasn't up to scratch. I don't know. But anyway, that methodology was something that I applied in the Virgin Islands. And now I can't get my slides move forward. Let's see. There we go. So I wanted to step back and give you a little bit of context of where I am. So the Virgin Islands is in the Caribbean. It's nestled between the Atlantic Ocean and the Caribbean Sea. And it's the teeny tiny, teensy little island that you can't even see, to the east of Puerto Rico and at the top of the Antillean island chain. And what I want you to really take away from this map is that each one of these islands has unique assemblages of species, of terrestrial species from the next island over. And that's because of geographic isolation over very, very, very long periods of time. So you have similar species on all of these islands, but they're different species, which makes the Caribbean a very high, a place that's got very, very high biodiversity. And so the animals that are there, the plants and animals that are there are really, really important globally because they're found no place else. They're not even found on the next island over. They're found no place else on Earth. So in the Virgin Islands, we have 5 native frogs, and they're all kind of just brown jobbies, except for this one, that's a brown and yellow jobbie, and this one's kind of greeny. And they're all about the size of my, this one, the Coqui is the smallest one, and that's the size of my pinky, my pinky fingernail. And the others are about the size of half of my thumb. So they're small, right? They're small little animals, but they make a lot of noise. We also have 3 non-native frogs, the cane toad, the usual suspects, the cane toad, the Cuban Treefrog. And then there's another frog, an Eleutherodactylus frog, from Puerto Rico that was not on the Virgin Islands when it separated from the greater Puerto Rican landmass about 20,000 years ago or so, at the end of the last Pleistocene. So cast of characters, right? The Brazilian is a cane toad, and I use these flags a la Olympic style to kind of allow you to maybe forget about the names and really think about where these animals come from. So the Brazilian is the cane toad, the Cuban is the Cuban Treefrog, the Puerto Rican is this little jobbie, this little brown jobbie that is so similar in ecology and size and diet and everything to the 5 native species of frogs in the Virgin Islands. So I'm just kind of out of interest, show you kind of size differential. These are to scale, right? Not actual size, but they are to scale. So these guys are smaller, considerably smaller than the native frog, than the non-native invaders. The native frogs are found really kind of across the islands in almost all the habitats. And this is what the Virgin Islands looks like, right? It's kind of dry habitat, dry forest, as well as some wet forest areas, some more mesic areas. The cane toad really needs water, needs deep kind of standing water and fairly limited in where it occurs. And the Cuban Treefrog is kind of found everywhere in forests and in urban areas, as well as the non-native, the Coqui, the Eleutherodactylus Coqui from Puerto Rico, are also kind of found in these human areas, but they're really closely associated with moisture in their environment. So one thing that's really important to understand about these animals, when you think about creating survey methodology for them, when we look for frogs in North America, We wait for the spring breeding season. There'll be that temperature threshold that triggers activity, and everybody runs out and they run to the nearest pond, and they make a racket, and they bring in their girlfriends, and they spawn. And so during that time, you can go out and you can find these animals, and you can conduct surveys for them when they're actively explosively breeding. And then for the period after that, you can look for those spawn, you can look for the tadpole, and then in the fall, you can look for the toadlets, the metamorphs coming out. In the Virgin Islands, however, and in many of the tropical areas, the frogs are direct developers because the tropics are characterized by very moist habitats without any discernible standing water. So the water is in the forest, it's in the trees, it's in the bromeliads, it's on the leaves, it's around, it's in the trees, the crooks of the trees and stuff like that, rather than being in ponds and streams. Or as well as ponds and streams. In the Virgin Islands, we don't have any ponds and streams. So you find these animals up in the trees. But what direct development means is that these animals are breeding continuously for the duration of the rainy season. And that sounds like a lot of work, and it is. And that's why the clutches are really small. So this is a single clutch for a frog. This is when they're first laid. And here's later on in their development, you can see that there's a frog in there. So here's a frog, there's its snout right there. You can kind of see back legs right here, that's its back, right? So they develop inside the egg capsule, and they're guarded by a parent who brings moisture in on their belly. And keep those eggs moist and guard the eggs from being eaten by other by insects and stuff. But the implications of this is that because the frogs are breeding across this long time period in the Virgin Islands, it's about 4 months, they are continuously calling. So you can do surveys at any time during that 4 months, and you can find them, and you don't have to go and look at them because they're everywhere. So oral surveys, so acoustic surveys, so listening surveys are great for detecting Virgin Islands frogs. Each frog has a specific call. You can hear them through the forest. They're continuously calling nightly, so they call for about 3 or 4 hours every night. And the active season is from May to December with a peak in September, October, November. What's not so great is that, you can only really find 4 of the 5 natives because one is silent. It's called a mute frog, and we'll talk more about that in a minute. You can really only detect 1 of the 3 non-natives. And that's because the non-natives have that temperate zone pattern of reproduction, and they call only when it's raining. So you have to go out on a rainy night to find those. Another thing that's not so great about the VI frogs is that they have a repertoire of sounds. So they don't just have 1 call that you can learn and follow. Each one has a range of different sounds that they make, that they sometimes make or sometimes don't make, or that frog sounds a little bit like that one, unless you really listen really hard. So it's really hard to train people to identify these different frogs. And so that makes it, that means that you either have to have someone who is willing to spend huge amounts of time to learn these calls and go out and listen for them, or it means that yours truly has to be the one that go out and do that all by myself. But if we think about sound, there's a lot of different information that we can get from sound if we're able to go out there and capture that sound. We can determine activity patterns. We can identify what the assemblages of species, and we can learn about who's living with whom, and who's active at the same time as others. We can follow the expansion of the non-natives. We can evaluate habitat use, response to disturbance, response to weather. And we can also, and this is the key one, we can also determine the patterns in human influences based on, what else is going on in that amphibian's environment, and whether that amphibian is still there after a house is built or a road is built, or not. And then something else that is really critically important but so underrated is the community interest. How interested people are in learning about and caring for these animals, because it's all well and good for your scientists to be out there studying these things and publishing in their scientific journals, but if the community of people that are actually doing those influences, those anthropomorphic influences, if they're not learning about what's going on around them, they're not going to care, and they're not going to do anything about it. So I moved to the Virgin Islands in 2004, and I followed the North American Amphibian Monitoring Protocol to do road transect surveys, to monitor activity and distribution. I did a study on Cuban Treefrogs. I did some chytrid testing, and I just did some kind of general schlepping around. Russ might recognize this as my game husband. Game on. He's so supportive and so interested and so keen to just kind of schlep around in the field, right? And by the way, Russ, this is actually Perseverance. This is the mangroves down at Perseverance where we took you that one time when we went out. And the amphibians, remember that picture that I showed you in Brazil, and I was marveling at how it's so small. These frogs really are literally everywhere, including on us, around us, everywhere. This is a baby metamorph or hatchling, I should say. So, okay, so the road transects, here's 4 years of road transects using oral surveys conducted 3 times in the active season. It takes about 2 hours to drive to get to 10 points. So I had 30 points, it would take a long time. And then you have to do it and everything. And one of the limitations is that part of my survey routes had to be removed because it was scary and it was dangerous, because St. Thomas is an urban island, and it's got the same kind of issues as urban, in our urban areas, including people with guns and the night that, I'm standing by the road and I'm listening and people come out of their house with their guns, that's when I was like, okay, not doing this anymore in this area. And, it takes a long time to drive. And again, I couldn't find anyone willing to learn the frog calls. So it's me out there by myself. But, you can get recorders. You can get recorders that you can put in place, and I have a bunch of these song meters from Wildlife Acoustics. Acoustic ones can, pick up sounds, sounds within the audible range of hearing, and then you can get some ultrasonic recorders that pick up, that have a specialized microphone that pick up bats and other ultrasonic organisms, whales. If I had a hydrophone, I could pick those up. But the limitations, there's limitations with these as well. They're expensive. You can only put them in one place at a time, right? You can't really, you have to have a lot of money to spread them around. There's cheaper technology. You can get little audio mouse, which is a little tiny Raspberry Pi microcomputer that you can weld on a microphone to it. You can hang those from trees, and they cost they're under $100 to build and program. But still, it's still pretty limiting in terms of scope. And just to remind you that even within the Virgin Islands, there's all these little islands. So logistics of getting around and actually getting those acoustic recorders out and positioned and then collected and maintained is really challenging. And then additionally, we have those islands over there, a different country. There's the US and the British Virgin Islands, and so there's jurisdictional boundaries, and then there's geographic boundaries, and travel isn't cheap, and there's all kinds of issues with permitting and all that stuff. So it's hard. So, but what if you could record across all islands at the same time, right? If you could do that, you could get all kinds of fabulous data and information, and you could look at some of those patterns I was talking about earlier, the distribution and the species assemblages and all of that. Well, I'm going to tell you that you can, and it's free, right? It's super easy, and you can just sit at home watching TV and eating ice cream while you're doing these surveys, and it's free. And that is because everybody has a cell phone, right? Everybody's got a cell phone, everybody's got voice memo on their cell phone. And so you can get everyone to go out and use their phone and take recordings and send them. And then you can just sit at home on your butt and listen to those recordings, right? So while I was training a student, while I was trying to get a student, I was trying to explain how to, survey for frogs, I thought, you know what? If I send the student out with her cell phone, I can get the same 30 sites that I used to do, minus the ones that are scary. Or I could just get everyone in the Virgin Islands to go outside during a short period of time, right? 2 weeks, during a really short time of the day when the frogs are most active, and everyone could collect cell phone recordings. So the pluses are there's no driving, no being in scary areas, no training, nobody has to learn how to do anything. Everyone has that technology right at their hands. And most importantly, there's access to all of those inaccessible areas, those private neighborhoods, those scary places. People live in those scary places. And, maybe some of those people that live in those scary places would be willing to go outside and take a recording, right? So there's potential for lots and lots and lots of data points. Risks, so negatives, people might not want to do it. So you might get some gaps in coverages, and people might not do it right. So you might get, bad recordings, and you might, something else. But who cares, right? Who cares about that? Because you can just throw those away. And in fact, I did get a lecture submitted. Somebody had recorded the lecture and sent that in, and thankfully it wasn't my lecture, somebody else's lecture, sent it in. I did not listen to that. So I created the grade VI frog count, and I had this thought on like a Wednesday, and I launched it on Friday without a whole lot of thought about it. And I had 4 very simple instructions that were a little bit complex in 2015. And I spent huge amounts of time talking people through how to use their cell phones, actually. But the simple instructions were go outside between 7 and 9 p.m. between the 1st and 15th of October, take a 2-minute voice memo recording on your phone, pin your location on your map app, because everybody's got that now, Google Maps or whatever. Just drop a pin and send the recording and the pin to this email account. And then I would spend the next year and a half downloading all those recordings and mapping those points and listening to the recording. Right, so the mapping part is pretty easy. You just open the drop pins, you capture the lat-long coordinate, you copy and paste it into a spreadsheet, you check the data against maps, tax records, people's site description to pinpoint the location. So you have to do some of that quality control of the data, always, 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 always. very tedious. And then you upload your spreadsheet into Google Maps or ArcGIS or some other mapping program, and voila, you have a map of where the recordings were collected. And I can actually do this during the 2-week survey period to identify those gaps in survey coverage, so that, you know, I can go out there, I can send somebody out there to those areas that are underrepresented and say, hey, you know, let's get some recordings from this area. So the first year, 2015, I thought, you know, when we first launched it, I was like, oh, you know, I'll probably get like 10 people submitting stuff. It'll just be my friends. Surprising to me, we got 317 recordings submitted. So remember, I could do 30 sites. This is 317 sites that were submitted from St. Thomas, which is my study site. This island here, St. Thomas, St. John. We got some from the British Virgin Islands. So these are the British Virgin Islands, and then from St. Croix. So this was the only island that I was really able to survey systematically, you know, on my own. And I was able to add all this additional data. And there's one, you know, kind of out there in the sea, which we ended up discarding because we couldn't figure out where that belonged to. So we did it a few more times, and I'll show you kind of subsequent years. But 2019, the last year that I kind of processed and we actually published this information, we had 181 submitted, and we added some islands, right? We added some of these islands in between St. Thomas and St. John. We got this island out here, Water Island, and we got some more St. Croix, and additional islands in the British Virgin Islands. So now we have data from 9 islands. So that's pretty exciting, really, that we can get that kind of reach. That's just the mapping, right? The frog ID part is a little harder, right? And let me just play for you a segment of one of these recordings. Oh, hang on. Did nobody tell me that I haven't shared my microphones? You can't hear these recordings, correct? No, sorry about that. Okay, so let me stop sharing. Share sound. Sorry about that. Okay, now tell me, can you? Sorry about that. Oh. Yeah, so that's what we hear in these recordings. I can't get rid of this. There, let's get rid of this. Throw that away. That's what we hear in the recording. I can hear 3, maybe there's a frog in there. There's also car noise, traffic noise, something else. So let me step back a little bit and tell you what we do with these recordings. So this is a acoustic analysis software program. It's called Kaleidoscope Pro, and you upload recordings into you upload audio recordings into the program, and it maps them. Basically, it graphs them. So on the y-axis is the frequency of sound. On the x-axis is the duration of sound, and then we have this amplitude segment up here. So the amplitude is the power of the sound, how big it is. The frequency is the frequency of the wave, and then the duration is how long it is. So each one of these green blobs, this is actually bats. But each one of these green blobs is a single vocalization. And by looking at the duration, by looking at the frequency range, we can identify what it is that's making that sound and distinguish it from other organisms that might be also vocalizing at the same time. So here is a wall of sound. I'm going to play this sound.
7:25, October 3rd, Lower East Carrot Bay.
Yeah, so that's Member Wheezy, right, who submitted that recording. So this wall of sound, and even within that wall of sound, I can distinguish different species, but I have to listen to it a few times. I can look at the spectrogram, so my graph of that sound, and I can identify, okay, so that's Coqui, that's Cochrane, and this is Antilensis. And I know I can just look at that and say, okay, I got 3 species in there, and this is what the species are. It's very, very quick and very easy. And you can filter out. So if I just wanted to look for Cochrane, I can filter so that I'm just got this range, this frequency range between like 3.5 and 4.5, and that's where Cochrane lies. And I can just filter all the recordings that belong to that, and I can create models for automatic identification of these frogs. It's hard because there's all this other noise. This is crickets. You get your anthropogenic noises down here, and it's a little bit challenging when you have a little bit of overlap in the species in there. But you can do it, and it's fabulous. And so I'm just going to play. So here's some frog calls that have been isolated. You can still see there's a lot of noise in there. And I'm going to just play this. So this is the Coqui, very simple call, very easy to distinguish. Right, so you have that 2-tone. And that what I just played is what's depicted in this graph right there. So that's really easy to identify. I don't even have to build a model. I can just go and look at all those. It's super easy to find. Oh, this one's a little harder. There's 2 species in here, and you have your Antilensis, has these two kind of tone calls, and then you have Cochrane on top of that. And then the Antilensis also has this reverb sound. And it looks like I have a question. Yeah, where Coqui got its name. Yep. That's why it's called Eleutherodactylus Coqui, because it goes Coqui. So let me just play this a little segment of this. Right, so that's that cheep right there. Did you get that? Did you hear that? Yeah, so super cool. And really easy to kind of, there it is, cheep. Really easy to kind of process these recordings. So let me get, blip, okay. Moving my Zoom things around the screen. So build models that sometimes work, sometimes don't work. And I also have written descriptions for each of the frogs, and I have protocols for the schools where classes, different age groups can do different types of activities with these. They can collect their own recordings. I have instructions for how to map them and how to analyze them using free software. you know, how to build an interactive map of the frogs, and then information about the frogs, who they are and what they do and stuff like that. So it's not just me that's learning about the frogs and what they're doing. I can also share this out to the people that are actually collecting these recordings. In theory, I could, if there was 400 of me. So we also, of course, do this with bats. And again, and I want to just go back here. So audible range is up to about 10. We can hear up to about 10 kilohertz. And the frogs are between, like, I don't know, 1.5 and maybe 4.5 kilohertz in frequency range. And I just want to show you the bats are between 10 and 40, or this one is between 30 and 60 kilohertz. So they're way beyond our ability to hear them. And we can still use this software to isolate those sound patterns and build models to identify these animals. Oh. Awesome. So there's a comment. Caroline Morrison is one of the VI frog count participants from North Star Village on St. Thomas. And I absolutely remember, because I process each one of these by hand, I absolutely remember your North Star calls. What's interesting about North Star, so I used to live with the pink whale, which is, so North Star is a place on the north side of St. Thomas. It's up high in the, you know, mountain, very, very wet habitat, very damp environment. And there's a lot of residential houses in North Star Village, but if you scooch over on the mountain a little bit to the pink whale, there are fewer residential houses. Still a lot of people that live up there. But North Star has all Coqui, all the invasive Coqui. There's hundreds of them, millions of them up in there, and another couple of native frogs. But if you go over to the pink whale neighborhood, it's the Coquis didn't make it there. They're totally different. Within not even a quarter of a mile away, you have a totally different species assemblage. And that is really informative as to how these non-native species are getting around. And of course, since I started this project in 2015, the pink whale neighborhood is now full of Coquis, and so that species assemblage has really changed. One of those native frogs has dropped out completely. I don't pick them up anymore in that neighborhood. So interesting. You know, I didn't bring any recordings of bats, but you know, we can, using the software, we can slow it down so we can hear it audibly, and they sound like a bunch of birds, kind of chittering, chattering, and it's really super cool to be able to listen in to you know, syllables and songs and sentences and language that we don't really understand. Okay, so frog distribution on St. Thomas, we have 5 species of amphibians, so the 4 natives and 1 non-native, and I think we also picked up the 4 natives and the 1 non-native. And I think we also picked up some of the Cuban tree frogs a little bit. So that was after the first year. We have a really good distributional range of all these different frogs. That was St. Thomas. This is St. John, which, you know, great areas of St. John are inaccessible except by trail, and so it's hard to get the surveys in there. But let me just return to this map of the road transects. This is what I had, just me kind of piddling around and stopping by the side of the road and listening, versus this, you know, with people's cell phones, right? So huge difference in the amount of data. And then you multiply that by all those other islands that we're able to survey. Okay, so invasives come and get blamed on irresponsible pet owners. We'll talk about that. I can talk about that. Remind me at the end and ask me that question. For sure, I'll talk about that. Because these are different invasives. These are different. This is a different thing. So, and some of this information, we can identify distribution where these animals are and how much. So these are the non-natives again, the Cuban, the cane toads, which are really restricted to areas where there's wetlands. The Cuban tree frogs are everywhere. Interesting Coqui distribution that's kind of related to the garden center. Right, the garden center in our side. So that's where the ag center is, where a lot of plants come in from Puerto Rico and are then distributed around. And the native frogs are all over the place. We can also see that there's some variation between site occupancy from year to year. So, these animals are not really moving around, but they are blinking. Site occupancy is blinking on and off, which could be based on localized, very, very, very localized weather patterns or habitat, or maybe some human disturbance, like a house was built or something. Right, and then we can also track the expansion of the non-natives across into new surveyed sites. So what can the frog count tell us? Not so much the daily, seasonal, and the activity patterns, because we're doing short surveys, synoptic surveys that are occurring at the same time, and so really hoping to really reduce the variation in weather across these sites. But we are learning a lot about about patterns in species distribution, where these guys are, where the non-natives are coming in and where they're going. We're looking at amphibian communities, and of course habitat use. Where are these guys and what are they doing? Kind of a little bit getting information on response to disturbance and a little bit weather influences. And certainly, absolutely getting patterns in human influences, because in the recordings, we also have other stuff. So I played that recording from you earlier. There was traffic in it. We're finding traffic, of course, because the surveys, if people are going outside of their house or they're driving around, they're going to be next to a road. And so many of the recordings have cars or traffic or something. Insects all over the place. We get dogs, we get people talking, music, TV, bars, which is really super interesting that you can have your frogs right in these, urbanized areas that, where there's not really a lot of habitat, and yet your frogs are still there. So that's really super cool. I also heard a cat on the recordings, 2 mosquitoes, several pearly-eyed thrashers, and a night heron, and I heard a bat drinking water. How do I know it was bat drinking water? Because it was my recording, and I was stood there, and I heard some ploppy plop 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 sound, and I turned my light on, and there was bat drinking out of a puddle, which was really super cool, and made me very happy. So subsequent, right? Remember that Virgin Islands had a hurricane. So I tried to use an app in 2016. So I started 2015, that really simple technology, just take a recording, email it to me. 2016, I borrowed an app and it didn't work. Crossed operating systems. I got so many emails and people got mad at me because they couldn't submit their recordings, and it just was disaster. 2017 post-hurricane, I tried really, really hard to get recordings, but they were all full of generators because we were without power in the Virgin Islands for 3 months without power. So generators, you can kind of hear the frogs, not so much. And then the other thing was, because we had no power, we also didn't really have cell phone so much. So it was really, really hard for people to actually submit their recordings. In 2018, I was like, oh, I can't do it. I'm tired. 2019, I went back to the original 3-step method. Take your recorder, take your recording, drop a pin and email it to me. I did the same in 2019. 2021, last year, you know, pandemic, I was tired. I didn't do it. I didn't do it. But 2022, I'm revved up, ready to go. Yeah, let's do the frog count again. Carolyn Morrison, you need to go out and move back to North Star. Yep. So I just want to kind of mention that in 2016, I gave a talk on this project at the joint meeting of ichthyologists and herpetologists. Jody Rowley was at my talk, and she's this charismatic superstar herpetologist in Australia at the Australian Museum of Natural History. And she and her team created a frog ID app where people are doing the same thing. They're recording frogs that they're hearing, and they're like chasing down the frogs. So you just submit whatever you're hearing, whatever frog you're hearing, and there's a team of people with, you know, headphones that will listen to those recordings and will identify what's in them. They don't so much do the mapping, but they do a lot more of that kind of outreach and information, and doing a lot of really interesting, cool stuff from the data that people are submitting. And she and I have talked about her project, and you know, she has $1 million or more and 1 million people working on her project. So she has this really super cool stuff that they can do. And it's really, you know, it was hard for me to get my study published because a lot of scientists are really skeptical of citizen science data and of the technology that everybody has. If everybody has it, then it must not be so great, you know, or whatever. And that's absolutely not true at all. That's not true at all. That's not what they found in Australia. That's not what I found. The recordings are as good, if not better, than the recordings off the professional equipment that's really super expensive. And so there's Jody, and she's a social media superstar. She's this great social media and media presence on the radio and TV and all those sorts of stuff in Australia. And you know, I can do the same too, right? There's me. I could be superstar glamorous in my non-glamorous self. But yeah, Great Via Frog Count 2022, first 15th of October, 7-9 p.m. We're gonna do it again. And I'm gonna, I just wanna make a note about iNaturalist. If you're not using it, you should be. It's a citizen science platform where you can submit your photographs, and as of maybe last year, the year before, you can also submit your recordings. So photographs of birds, bird calls, frog calls, all that stuff you can submit to iNaturalist. And I just want to say that it's the data that citizen scientists are generating. So citizen scientists are people in the community that are non-scientists that are collecting data, and in many cases are analyzing the data as well, along with the scientists. It's so valuable, not just to the scientists, but to the people that are doing it, in terms of raising awareness of what's around us and why we should care about it, and just how fabulous things, you know, how fabulous our natural world is and how frighteningly quickly it's changing. Okay, so the Virgin Islands mottled frog, the mute frog, ever was thought that it was silent. It's silent. The theory is it originated on St. Croix where there's no other frogs and it was all by himself, and it didn't need to have a call. But what I discovered in the 18 years or 20 years, whatever, that I've been studying frogs, is that they are, and if insomnia, right? Because I'm lying on my sofa surrounded by frogs, in my house, and not in the house, but they're outside the house. What I discovered is that these frogs are calling in the middle of the night when the other frogs are silent. And so I can, let's see, kind of go to the next. Yeah, no, I'm not gonna play. Well, all right, it's right at 1 minute. There it is. Did you hear it? There it is again. Did you hear it? So what's super cool about that is that we have that signal on that. I dug out that recording, found that signal, and I'll just play you this little segment and you can look at it. Right? Did you hear that? Tick, 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 tick. So that has a very soft, you can see how it's really, really hard to see. It's hard to hear. And it's just not a call that people are going to hear. And indeed, this was the one recording where this came up, and it was a recording that somebody had taken, after a night out in the bar. He got home at like midnight, like recording the frogs, wrong time window. Everything was bad about this recording, and yet we got really valuable information out of this recording from this person that didn't follow the directions. So even when it's wrong, it's still fabulous. And that's its signature right there. Tick, 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 tick. And that's really all I have to say about this project. This is me and us. We have Facebook presence for VI Frog Count, VI Bat Team, and Virgin Islands Wildlife. Frog Thing has a really crappy website, but it's a website that I built, and you can go and get more information about it. Yeah, so any questions? Okay, so Russ had a question about non-natives, and I did a lot of work in England. I did my PhD, my master's and my PhD in England, and I established the Kent Reptile and Amphibian Group, which aims towards recording the distribution of reptiles and amphibians across the county of Kent in England, in Southeast England, and where all natives, everything. And then one day some of these other organisms started turning up. We started getting pool frogs, we started getting wall lizards, and we started getting some other weird, animals turning up. And in recording distribution of some of these organisms, they came from one kind of area, which correlated to, and it didn't just correlate, we tied it right in. His guy named Lee, who was a herpetoculturalist, and he was letting these things go. And now the wall lizards from the Mediterranean are established in the UK. There's a lot of, there's kids that are breeding things and letting them go because they think it's good for conservation. And it is not good for conservation. It is not good to have these non-native organisms out because they have all kinds of impacts on the native guys that we're trying to protect. And what your Cuban tree frog does in one place is completely different from what your Cuban tree frog is going to do in another place, depending on the species assemblages and the habitat and stuff like that. So it's very problematic, and compounded to that is climate change, and the fact that these animals, many of these animals, are moving themselves to new locations because they can no longer survive in their, in their usual habitat, in their home habitat, if you like. And so our thinking as scientists, we can't protect our biodiversity. We can't do it. It's hard to accept that, but with all of the human changes to the landscape and to the climate, we are not going to be able to protect our species assemblages and all that great, yay, native guys. And really, we're coming around to thinking about what is functioning on the ecosystem. We want functional ecosystems, and so that does actually mean, so the Coquis are coming in and they're out competing all of our native frogs, and it's heartbreaking, but at least we still have a frog on the landscape. Right, and we have a more robust frog on the landscape because it's a better competitor than the native frogs. So we say goodbye to the natives, but we still have a frog, and we still have a functioning, frog in the landscape. And the frogs, the non-native frogs in the Virgin Islands have come in not from the pet trade. None of these are pets. They've come in from like in landscaping material and in construction material, and just as hitchhikers, right? Snakes will crawl into boxes, and those boxes get put on a cargo ship, and the snake survives. So we have a good population of corn snake in the Virgin Islands. Yeah, many invasives get to the new areas through global economy. Absolutely. Yep, they're transported in boxes, they're transported on cargo ships, they're transported in people's suitcases by accident, bugs and stuff. Yep. So any other questions? All righty. I'll shut up.
Well, thank you so much for joining and sharing. It's definitely been a pleasure. Looks like one more came in. There was an ask about bats recovering after Irma and Maria. Caroline was saying she recalled a few species that you had been concerned about.
Yeah. So if you're unaware, the Virgin Islands, the little, tiny little islands, and in 2017, we had 2 Category 5 hurricanes that were ginormous. They were, the size of France. Irma was the size of France, and it took out St. Martin. It took out the British Virgin Islands. It took out St. Thomas. It went north of St. Croix, north of Puerto Rico. And it was Category 5. It flattened everything. And in flattening everything, it, took out trees and leaves and habitat for everything, and the bats just couldn't find anything to eat. They died. They were dying all over the place. We were hanging out bananas and all that. 2 weeks later, Maria came through, which is also a Category 5, and it had a little bit of a southerly track, so we got clipped by it, but it took out St. Croix and then Puerto Rico. And so the bats that were able to escape Irma and found their way to St. Croix and to Puerto Rico were then then had to deal with the aftermath of that other storm. And so there was no fruit, flowers, anything for those animals to eat for a good 3 months before the trees started producing flowers. So It was a couple years. We started doing, I mean, we've been doing monthly surveys for bats since I don't know, forever, since 2008. We've been doing monthly surveys, and we continued doing those as soon as we could get back to our study site. And we caught nothing for months and months and months. And then we started getting a couple of things. And it's been, I guess, three years after Irma, we started seeing reproduction. Yay! We started seeing reproduction. And so now we're back to still lower numbers of the bats are really common. We're still seeing lower numbers, but we are seeing reproduction. We're seeing pregnant females, lactating females, and young of year pups flying. What we have not seen, and this is, I guess, your question, we still have not seen the Stenoderma rufum, the endemic little, sweet little poor flyer bat that needs to live in forest all by itself. We're very shy, very habitat specific. We still have not seen, we had recorders all over St. John looking for them. We've had recorders all over St. Thomas looking for them. And I've spent thousands of hours. I've gone through 500,000 recordings looking for that vat, and we haven't found it. I don't think it's gone, ever gone, just can't find it.
Maybe up in El Yunque.
Sorry?
So maybe up in El Yunque.
Yeah. Well, you know, what's interesting is they had done a lot of surveys at El Yunque area. I remember Hugo went through there. And so they had before and after surveys at El Yunque. And that bat disappeared and never came back from El Yunque. Disappeared from there and never came back. So, you know, I don't think they're gone. We just don't know where they are. Yeah.
Well, it's good to see you, and thank you.
Yeah, thank you. I'm surprised that you're on here. I'm delighted. Yeah.
Well, I saw your name. It's like, oh my gosh, I have to come in and hear the update on the frogs. So
awesome.
yeah, it's worth the trip down there just to go out at night and just listen to all the night sounds. If you're for frogs, it's just amazing.
Yeah.
And look at the stars.
Yeah.
Thank you, Dr. Platenberg. That was really awesome.
Yeah, awesome. You're welcome. Thanks for inviting me. I appreciate it.
No.
You're quite welcome. It's our
pleasure.
pleasure.
Thanks for joining us, and thanks for everybody for coming out and sticking out. It's always great to continue to share and grow and all of our unique knowledge. Bronwyn wanted me to remind you all, raffle this month is a crab feast kind of take home event. So if you're interested in that, check it out. It's $5 a raffle ticket. We'll pull a name and somebody wins like a bucket of crabs and some beers and some other things. I'm not exactly sure all the good stuff in it, but if you should check out your email, because I'm sure you got an email about it. If nobody else has questions, I'm gonna close out for the night, but thank y'all for coming, and hope to see you all again soon.
Thanks. Good night.